Meenakshi ma'am, we are live now. You can start. Thank you.
Thank you, Anil. Do you know that your baby is able to communicate with you long before he or she speaks a single word? A baby's cry, smile, vocalization, gestures, imitation, eye movement, gaze, eye contact are all part of their communication. All these skills are very important and lays the foundation for developing speech and language and helps a child to talk and communicate. If you find that your child is having difficulty with these pre-verbal skills, please consult a nearest qualified speech language pathologist at the earliest because early intervention, early help can prevent difficulties with the verbal language which the delays that can happen later in the child's life. Good evening. I'm Meenakshi Vadhera, an audiologist and speech language pathologist. And on behalf of Indian Speech and Hearing Association, I welcome you again to another educational session on of Speech and Hearing Annuals 2020. The sole purpose of these annuals is to spread awareness about various kinds of speech, language, or hearing problems, various aspects of these problems, and how they can get the best possible help from a professional. But before I start, I would like to invite Mr. Suman Kumar, who is the chair of professional matters of Indian Speech and Hearing Association. Suman sir.
Thank you, madam. Uh, Good evening to one and all. On behalf of Indian Speech, Language and Hearing Association, I welcome Professor Sri Salapati Madam from University of Madras, Professor Anand Sir from JNCASR Bangalore, and Dr. Sudha Maheswari from MERF Chennai for today's panel discussion, that is deciphering the role of genetics in communication disorder. I also welcome Rangasai sir, former director of Alia Virgin National Institute of Speech and Hearing Disability, NIEPID Sikandrabad, NILD Kolkata, and CRC Ahmedabad and CRC Bhopal for moderating this important session. I think today's discussion is going to be very important because we all know that developmental disabilities have a genetic cause, and if these are identified and taken care, then we can avoid future complications. So without much delay, I request Rangasai sir to kindly take over and proceed.
Thank you, Suman sir. Before we start, yeah. So, as sir has already told us that we are going to be talking about genetics. Genetics of deafness and speech language disorder is increasingly throwing greater knowledge and insight into managing communication disorders, the development in gene based intervention, and efforts in stem cell therapy have been increasing our hope in curing these disorders. Let's listen to what our panelists have to say. But before that, so we had yesterday we had a very informative session on autism, and if you have missed this session yesterday, you can still watch that on our YouTube channel called Isha Annuals. And tomorrow we would be having another session on cochlear implants and how they can help a child or an adult. So today we are going to put the facts right. So to moderate this session, we have with us Professor Rangasai Raghunathan Rao. He is the chairman of Audiology Committee of International Association of Logopedics and Phoniatrics, which is at Malta, a federation audiologist and speech language pathologist of 44 countries in consultative status with WHO. He is a member of ICMR's Task Force on Deafness. Formerly, he was a technical director and professor at Dr. SRC Ish, Professor AYJ, NIHH, NIOH, NIMH, CRC, and member of NBIS. He's the founder member of the IEE Disability Group, Bangalore, and has been the president of ISHA two times, one from 96, from 1986 to 87, and then from 14 to 15. And he's been also the founder president of ICAA India chapter. He has extensive experience in serving delivery in rural areas and immense interest in empowering people. He has numerous awards, researches, and publications to his credit, and he has always been very, very helpful and always come forward to help not just the professionals but to the public wherever possible. So, our next and that the whole panel constitutes of very elite panelists, which are masters of this topic. So I would like to first introduce Dr. Anuranjan Anand. He's a professor, as Suman sir has already said, he's a professor of molecular biology and genetic unit and chair and adjunct faculty of neuroscience unit at Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. He is a PhD of microbiology and cell biology from Indian Institutes of Science, Bangalore, and postdoctoral from Department of Biological Studies, Stanford University, California. He has numerous fellowships, and which includes a fellowship from Indian National Science Academy, Delhi, fellowship from National Academy of Science, Allahabad, and Indian Academy of Science, Bangalore. He's got a National Bioscience Award from Department of Biotechnology, New Delhi, and also a Outstanding Research Investigator Award.
Thank you, Meenakshi.
So it's my privilege again to introduce Professor C.R. Srikumari Sri Selapathy. She's a UGC research scientist at the Department of Genetics, PG Institute of Basic Medical Sciences, University of Madras. She's a PhD in human population genetics, postdoctoral fellow in molecular otolaryngology from University of Iowa, USA. She's taught in Aish, Mysore, our own institute, and at AWH Calicut. She has 10 PhDs and 30 MPhils with emphasis on Fragile X and genetic deafness. Has about 76 papers published and 3 book chapters. She currently is working on genomics of hearing impairment, a large cohort of assortative mating, and constructed repository of DNA database for deafness and stuttering with genetic register. Thank you, ma'am, for being with us today. And now the next panelist is Dr. Sudha Maheshwari. She's a consultant at Madras ENT Research Foundation, Private Limited, Chennai. And she's the head of the Department of Community Otolaryngology at MERV and in charge of the cochlear implant program there and under the Chief Minister's Comprehensive Health Insurance Scheme. She has 14 years of clinical experience in the management of children with congenital, non-syndromic and syndromic hearing loss. She completed her doctoral research work on the genetics of hearing loss in South Indian population and was an invited faculty at the World DNA Day conference held in China. She's a recipient of Dr. Sandadisa Gold Medal, the AI South Zone Gold Medal, and she has also received the International Visiting Scholarship Award from the American Academy of Otolaryngology. Thank you, ma'am, for joining us today. Over to you, sir. Dr. Rangasai, sir.
Thank you. Thank you very much for your kind introduction. Dear all, I wish to share with you few messages, anecdotes, and concepts that are pertaining to the theme of today's webinar. In our conversation and discussion, we may, in view of the heterogeneous audience that we have, I will request you to forgive in the event we use terminologies that may not really be acceptable to you. We have taken care not to be saying any such, but however, there is no, our intentions are supportive. So that's a point that I wish to make before we start. Today is World Human Rights Day with the theme as Recover Better, Stand Up for Human Rights. Be that as it may, the Article 15 of Constitution of India talks of prohibition of discrimination on the grounds of religion, race, caste, sex, or place of birth. In this context, in today's context, it makes me think that probably the word race also includes genetic variations among us. So, kind of, I just want to fit in that meaning in this context here, and then see how we may take it forward. Today, glad that we have gathered on this platform to discuss about genetics of communication disorders. Genetics is the study of how genes bring about characteristics or traits in living things and how those characteristics are inherited. The word heredity or genes is not new, even in our colloquial language. In fact, we had a lot of discussion, how would people understand the word gene or heredity and all. So in that context, I add this, that when someone wants to describe us, they use, okay, he is from that family, this family, and he is tall like his father, or it's all in her genes. She has musical skills of her grandmother. Even in colloquial terms, this concept that we inherit few things is not out of place for any common man to understand. You will get to know from our eminent panelists on genetics of communication disorders. Personally, I got introduced to genetic testing as a clinical procedure in late 1970s by no less a person than Dr. Marimuthu as uh, as head of Postgraduate Institute for Basic Medical Sciences, Karamani. The team of scientists headed by Dr. Arbandi Ramesh showered with a lot of knowledge and the details of chromosome testing at that time and drawing family tree. So this is what exposure we got at that time. Later, in 1985, when I went into the district rehabilitation scheme in the villages, for example, in Kattanguluthur block in Chengalpattu district, we found a lot of families with problems. But there was no genetic testing or counseling facility at that. Believe me, at that time, there were only two disciplines that studied genetics. One is persons doing MSc in anatomy. Anatomists were studying genetics. And the other one happens to be a veterinary doctor. So in fact, we partnered with a veterinary doctor to provide based on pedigree drawing for the purposes of this. This was the state at that time. Later, in late 90s, Professor Vijay Raghavan, who was then with Indian Institute of Science, Bangalore, and NCBS, National Center for Biological Sciences in Bangalore, and now Science Advisor to Honorable Prime Minister, introduced me to one of the panelists today, Dr. Anuranjan Anand. We as a team of professionals that includes Dr. Shelly Chadda, now with the WHO, and Dr. Arun Agarwal, former Additional Director of Director General of Health, both from MAMC, New Delhi. We turned out a lot of work in this area and publications as well. I shared these brief encounters with significantly important people today only to flag that we are in a country and at a time period when we can usher in collectively a lot more in this sector dealing with genetics and disability, especially with Rights of Persons with Disabilities Act 2016 being effectively implemented. Now, I'll shift to another concept. You are all aware that the International Classification of Functioning Disability and Health, in this framework of WHO, the body structures and functions, activities and participation in the context of environmental and personal factors are important. So genetics in essence deals with body structures and functions. So what we are today discussing falls well within the framework of we understanding how to go about and what is to be done in our own framework. I wish to highlight that ICF considers attitude as an important environmental factor that may act as a facilitator or a barrier to stakeholders. Underlying point is attitude matters. So that is something that I want to underline in this structure. Then, normal development of communication requires the interaction of an intact mechanism with a favorable environment. The components of intact mechanism include hearing sensitivity, perception, intelligence, structural integrity, motor skills, and emotional stability. As part of auditory system, we also have, we as audiologists also address issues such as vertigo and tinnitus. Okay, be that as it may. Few years ago, the National Institute of Health had sent a communication where they indicated that the hearing system is governed as many as about 3,000 genes. Okay, needless to say here, how many more can be there related to perception, intelligence, structural integrity, that is to speech as a complex activity. At this juncture, I also want to add that the future, the medical profession, how it is moving ahead in the health sector, as per NIH director's report, it is predictive, it is preemptive, and it is personalized based on the genomic aspects. All the three are based on genomic perspective. Predictive, what all mutations, wrong mutations or their negative mutations, all that could be identified. And what are all preemptive, meaning removing the predisposing cellular factor from the genes. So this is preemptive, it is not preventive, not giving a vaccine, but it is the cellular basis, biological basis for getting that is removed. So this is preemptive and personalized because each person's biology. So this is how it is moving towards. So again, I wish to inform you that what we are discussing today is very, very relevant and leading. Reportedly, there are over 6,000 genetic disorders. Okay. We use the word called mutation. I just wish to share for the larger audience. A change that happens in genes, mutations or a change that happens in genes are one of the fundamental forces of evolution enabling evolutionary change. Based on their effects on fitness, based on the effects of these changes on fitness, there are three broad categories, namely advantageous or good genes, good mutations. That is, that increases the fitness. The deleterious or bad mutation that decrease the fitness, and neutral ones. Okay, so this is the fundamental concept in a language which is as simple as possible. I've tried to give, but you will be getting, listening to all the experts. The details with respect to communication disorders will be addressed today as comprehensively in a very understandable word. I request you to kindly note down whatever you want to ask. We know that we are entering into new terms, terminologies. Therefore, we got to be, I request you to kindly be more attentive in all this and note down, and in question answer session, we will be able to answer. With these few words, may I start with the first question. This is one question. The question is, can you please explain basics of genetics? This is addressed to Dr. All the three panelists will be answering. First, it will be Dr. Sudha.
Thank you, sir. So I'll share my screen. So thank you, Madam Meenakshi, for the kind introduction, and my heartfelt gratitude to the Indian Speech and Language Association for such a fine opportunity. So let's just refresh the basics as far as we can. So maybe I'd like to familiarize the audience with some of the basic terms we use in genetics. The first thing is a gene. What do we mean by a gene? It's the basic unit of inheritance. And the genes are nothing but DNA sequences which are organized in chromosomes. And the nuclei of all human cells contain 46 chromosomes, which are organized in 23 pairs. We have autosomes and allosomes, sex chromosomes. And the sex chromosomes, the genotype is XX in females and XY in males. So this most of us will be familiar with, just to refresh a bit of the basics. So what does the term genome means? Genome means it refers to the collection of all the genes which an organism possesses. And in the human genome, there are approximately 3 into 10 to the power of 9 base pairs of DNA, which makes up the haploid genome. And DNA, or the deoxyribose nucleic acid, is basically made up of many nucleotides. So it's actually consisted of polynucleotide chains. And what is the structure of each nucleotide? Each nucleotide consists of a sugar, and in DNA, it's a deoxyribose sugar, a phosphate group, and a base. This is the basic structure of a nucleotide, and many such nucleotides form a DNA molecule. And the bases which are present in DNA are of two types. We have the pyrimidine base and a purine base. Basically, both are structurally different, in which the pyrimidine is a smaller one containing a carbon ring with 2 nitrogen atoms, and purine is a double ring structure consisting of carbon molecules, hydrogen, and 4 nitrogen atoms. And the purine bases present in DNA are adenine and guanine. And the pyrimidine bases, we have cytosine and thymine in DNA, and uracil in case of RNA. And it is the presence of this thymine in DNA is what makes the DNA a more stable genetic material than RNA. So most of us might have that question, why DNA is a good genetic material? Why not RNA? It's because of the structural stability and the presence of thymine, which makes DNA a more stable genetic material. So that's the universal blueprint of life on Earth. It carries the genetic information, passes it on from the parents to the offspring. So this is again the structure of DNA molecule. It's like a spiral staircase. So this is a polynucleotide chain, and this is another polynucleotide chain. So we have 2 polynucleotide chains. Each polynucleotide chain is made up of numerous nucleotides. The basic nucleotide structure we have just now seen, it's made up of a pentose sugar and a phosphate group and a base. And these 2 chains are always complementary to each other, in that the purine base is arranged in such a way that it can pair with a pyrimidine base. So it's actually called as complementary base pairing. Adenine always pairs with thymine, and cytosine always pairs with guanine. So in a DNA molecule, the number of adenine bases is always equal to the thymine bases, and the number of cytosine bases will always be equal to the guanine bases. So it's like that. And adenine pairs with thymine by means of 2 hydrogen bonds. Cytosine pairs with guanine by means of 3 hydrogen bonds. And these hydrogen bonds are not as strong as covalent bonds. So during replication, what happens is we have these two DNA strands, and these strands separate. So one of it acts as the template strand from which the RNA copies the information. So this hydrogen bonding enables easy detachment and replication. It's not a very strong bond. So this sugar phosphate bond forms the backbone of the DNA molecule, and the purine base in this strand pairs with a pyrimidine base in the other strand. It's vice versa. So this structure offers more stability to the DNA. It becomes a more effective genetic material than RNA. And the transfer of this information from parents to offspring, it is called as inheritance. So what characters we inherit from our parents, and the transfer of this information from the genome within the nucleus. So the nucleus contains the chromosomes, which contains the DNA. Okay, and from this, if the information is passed on to the cytoplasmic level and where it forms a protein, this is called as gene expression. And this occurs by two processes. One is called as transcription. The other one is called as a translation. And what is a gene coding sequence, and exons, introns, start-stop codons we'll be discussing in the further slides. So what is actually a codon? A codon actually is a triplet word. So we have seen that there are about 4 bases in a DNA. We have adenine, guanine, cytosine, and thymine. If these 4 base pairs are arranged in orders of 3 letters, 4 to the power of 3, we should be having 64 codons. And each codon actually codes for a particular amino acid. So we have only basically 20 known amino acids. So we have one or more codons coding for more amino acids. But instead of having 64 codons, we have only 61 codons coding for all these amino acids, because 3 of those sequences, which is called as UAA, UGA, and UGG, they do not code for amino acids. So they are called as stop codons. So what do we mean by stop codon? That doesn't code for that particular sequence. It doesn't code for a protein. That we call it as a stop codon, and which initiates the coding, that's called as a start codon. And exons are regions in the DNA which codes for the protein, and introns do not code for a protein. Introns are the non-coding regions. And as I've told, there are 2 main processes on passing on the information. One is the transcription, in which the messenger RNA copies the information from the template strand. And the second process is called as translation, in which by the copied information, it gives information to form a protein at the cytoplasmic level. So this protein affects the cellular function at different levels of the body. So this is the central dogma of molecular biology. So we have seen what is a gene. Gene is nothing but a DNA sequence, and it has too many codons, actually 61 codons. It can be arranged in letters like AGG, AAA, like however it is. There are about 64 sequences like that, of which 3 sequences do not code for any protein, and we have 61 codons coding for proteins and making the cell function. So here we have the cell, the nucleus, inside which we have the chromosomes, which contain the DNA and the genetic material. The messenger RNA takes the information from that by a process of transcription, and it forms a protein. So this is called as translation. So how do genetic disorders happen? Any disruption in any of these processes results in a malformed cellular function. Okay, so that's the basis of a genetic disorder. And that's it, sir.
Okay. May I now request Dr. Sri Kumari, please.
Thank you. A firm basis was formed. Before I join the discussion, I wish to also thank the organizers in Isha for this opportunity that allows us to share the essentials on heredity to the community at large. I also wish to spend a moment to take the name of Professor Ramesh, who is indeed the founder of deafness genetics in India and the former professor and head of the genetics IBMS. All my co-panelists, including me, had the good fortune to work with him in hereditary hearing impairment. He began 3 decades ago and proposed the first two locus multiple recessive homozygous genetic models studying the South Indian hearing impaired. Of course, he was quite ahead of his time because the model was proposed when not even a single gene was identified for hearing impairment across the genome. Dr. Sudha has set a foundation and she has given technically about the meaning of a genome and its architecture. So let me give you a simple analogy since I've also been asked to comment on the basics of genetics. Uh, for the layman, it can be understood as a genome as a book. In this, there are 23 chapters called chromosomes. Each chapter contains several thousand stories called genes. Each story is again made up of paragraphs called exons, and these stories are interrupted by advertisements called introns. Each paragraph is made up of words called codons that was well explained just now, and each word is written by letters called bases. So, of course, one more important thing is genome is a clever book because it can photocopy and also replicate. When genes are replicated, mistakes are sometimes made. A letter or a base is occasionally missed out, or the wrong word or a letter is inserted, as in a text. Whole sentences or paragraphs are sometimes duplicated, omitted, or reversed, and these mistakes are known in genetic jargon as mutations. So the codons also were well explained. And please note that this is a simple concept in nature. These errors or mutations can sometimes drastically affect an individual's communication and can be inherited. We call combinations of such mutations as pathogenic phenotypes, and the manifestation of the genotype as phenotype. Having said this, when you look at the number of genes allocated for these communication disorders, the lists are going to be long and variable. More than 150 genes have been discovered to cause hereditary deafness. And then there are two types of hearing impairment. One is the non-syndromic and the other is the syndromic. And these are the number of genes that have been implicated. Moving on to the speech and language, there are about 48 genes that have been implicated in language disorders and 39 genes in speech disorders and 19 genes that have some association with speech language deficits and complex syndromes. So they are actually called as potentially involved. Coming to autism, 100 genes have been strongly associated to autism. And since cleft lip and palate also has something to do with the communication, I have added that in the list. And there are 40 genes that have been identified for both syndromic and non-syndromic cleft lip palate. So this is the expansive list that I thought I would bring to you to only impress upon you the genetic heterogeneity. Yes.
Thank you. Thank you, madam. Professor Anand, please.
So please unmute yourself, sir.
You are not being heard. To your
Sir, please switch on your mic, sir.
yes, no? Yes, done.
Are you getting me there?
Yes, very much sure.
Yeah, I'm sorry. I couldn't unmute myself. Um, don't know the reason. Now, let me start by thanking the association for giving me this opportunity to present our views. Uh, Meenakshi Vadhera madam, all the other co-hosts, and Rangasai sir, and obviously other panelists, uh, Sudha madam and Shikumari madam, for introducing the topic before me. Now, I want to give a small twist uh, to the story of basis of genetics, because you've heard uh, some structural aspects of the genes in context of the human genome size, etc., etc. But the genes find their basis of action, which is embedded in the auditory structures and cells. Uh, Cell being the most basic unit of formation of any organ structure, including auditory ones. Uh, Development of these cells and structures from the embryonic stages to the adult stages, they're functioning throughout their life. In the context of today's discussion, uh, cochlea being the main signal transduction apparatus, um, hair cells, supporting cells, and all the structures that you can see on this slide. I won't go through naming each one of the cell types or tissue types that you are familiar with, are the ones which give the meaning to the genes, because genes have to function in their formation and have to ensure that they continue to function throughout our life in a meaningful manner. As we move forward and talk about genetics and associated things, I'll also try to elaborate um, how we are trying to ensure, trying to facilitate what kind of research is going on in these angles. So for the time being, I'll stop here with the basics of genetics and what do genes do. They essentially build up the structure of our, auditory processes.
Thank you, Dr. Anand, for giving such a concluding remark. I think we'll go to the next question. What are types of genetic disorders? Dr. Sudha, please.
Yes, sir. Sure. Thank you. So among the genetic disorders, sir, we have chromosomal disorders, which are again classified into numerical and structural abnormalities. Numerical abnormalities, many of us will be familiar with, conditions like trisomy 21, Down syndrome, Klinefelters, Turner's, and chromosomal abnormalities with structural disorders and monogenic disorders, in which only a single gene is implicated in a genetic disorder. And we have complex disorders in which more than one gene is involved in a condition. And this, Madam Shikumari has already given an intro about this, like mutations is nothing but a change in the genetic sequence. So we have seen about a normal genetic sequence, and any change in that sequence causes a change in the function. So that's what is called as a mutation. And mutations, we have two types of mutations, point mutations and frameshift mutations. And point mutations are actually very simple mutations. Otherwise, they are simply called as substitution mutations. Suppose if there is a base, for instance, this picture shows adenine. If it is replaced by cytosine instead of adenine, so there is a change in the base. So this is called as a point mutation, in which the change occurs in only one place. And again, point mutations, we have two types. We have a transition or a missense mutation and a transverse or a nonsense mutation. So what happens in a missense mutation is, so we have went through while going through the basics, we have two types of bases, purine bases and pyrimidine bases, right? So if a purine base is replaced by a purine base, or a pyrimidine base is replaced by a pyrimidine base, then it results in a missense mutation. And as a result of this mutation, what happens is the genetic sequence is changed. So the resultant amino acid that is formed is changed. So the resultant protein is altered. So this is actually a missense mutation. What happens in the nonsense mutation is a purine base will be replaced by a pyrimidine base and vice versa. In case of pyrimidine, it will replace purine. And what happens is the resultant codon that is produced results in a stop codon. So the protein is not formed. So the resultant protein is a short or a truncated protein which is malfunctional or it doesn't function at all. So of these two mutations, nonsense is a little serious mutation. And what do we mean by this same or silent mutation? So while refreshing the basics, we have seen that we have 61 codons coding for 20 amino acids. So we may have 3 or more or even more codons or sequences coding for the same amino acid. So there will be a change in the base pair, but the resultant amino acid that is formed may not be altered at all, because the changed codon also will be producing the same amino acid. So there is a change in the base pair. There is a mutation which we'll be able to see, but that doesn't cause any change in the protein or that doesn't alter the function. That is called as a silent mutation, which is a harmless one. Coming to the frame shift mutations, these are mutations in which more than a single base pair is altered. Either they are inserted, they are deleted, and this results in the complete change in the reading frame of the genetic sequence. This is a more deleterious mutation when compared to point mutations. And the patterns of inheritance, we have four primary patterns of inheritance: autosomal dominant inheritance, autosomal recessive inheritance, we have X-linked inheritance, and mitochondrial inheritance. So we'll just go through in brief, all of these. A dominant hearing loss is caused by only one copy of a mutant gene. So all of us have two copies of a gene, one inherited from father, one inherited from mother. For a dominant mutation to produce a disease, only one copy of the mutation is enough. That means even in the heterozygous state, even when the person has an other normal allele, then they'll be able to produce a disease. So this is actually an example of a dominant pedigree. And in this, actually, every child of a parent who has a dominant mutation has a 50% chance of inheriting it. And when coming to the autosomal dominant inheritance, almost in every generation, we'll be seeing the disease. From the parents, it will be passed on to every generation, and each child will have a 50% chance of inheriting it. Autosomal recessive inheritance, on the other hand, we need both the copies of a gene to be abnormal or mutated to get hearing loss. If only one copy is present, then that person will not manifest the disease. That person only will be a carrier of the disease. And if the mutated genes are present in the homozygous state only, then the disease will get expressed. And in these conditions, mostly, as we can see in this pedigree, the parents will be normal, unaffected. And each child will have a 25% chance of having the disease, a 25% chance of being normal and a 50% chance of being a carrier of the disease. They'll be only carriers, they'll not manifest the disease. And the autosomal patterns of inheritance, dominant and recessive, they are all linked to the autosomes or the somatic chromosomes. And the X-linked disorders are related to the sex chromosomes. Here again, we have a X-linked dominant disorder and X-linked recessive inheritance. This is a classical pedigree of a X-linked recessive inheritance. And here, only males will be affected because the male genotype, we have XY because there is no other normal functional X gene. So all the sons who inherit this affected mutated X will manifest the disease. So each son will have a 50% chance of having the hearing loss. Each daughter has a 50% chance of being a carrier. And daughters will mostly be carriers and males will be affected in X-linked recessive inheritance. So this is a classical pedigree of a mitochondrial inheritance. And mitochondria, only the mother passes a mitochondria to her children because during fertilization, what happens is the mitochondria that is present in the sperm tail will get destroyed. And so the mitochondrial inheritance is only from mother, but all the children of the affected mother will be affected because it's passed on through mitochondria. So that's it, sir. This is about my types and patterns of inheritance in genetic disorders.
Thank you. Uh, can I ask Dr. Sri Kumari, what is the pattern of single gene inheritance? Am I heard properly?
Yes, sir.
Okay,
Sir,
thank
you are
you.
logged in using two devices.
Yes, because I had problem with that. I'll now switch off. Thank you.
I have heard you, sir. Actually, Dr. Sudha has given all the patterns of inheritance, and this is a summary slide that once again brings all the patterns of inheritance. So I'll just skip this and go forward to illustrate the patterns of inheritance in a real time situation. This is something we obtained during our laboratory analysis. This is a connexin gene mutation, and we have a family history that can itself provide diagnosis at times about the patterns of inheritance. So this is a medical pedigree, which is a graphic presentation of a family's health history and genetic relationships. All health professionals, including speech language pathologists, should learn to depict a pedigree in the standardized format and should keep that as one of the data sheets in their case reports. This is what I wanted to emphasize. Now let us look at the right hand side of the slide where I have depicted an autosomal recessive pedigree. Notated on the left side of the pedigree is the generations. This is the standardized way of representing a pedigree. So we have the first generation, second, third, fourth, and fifth. Across the five generations, you have some double bonds being looked at. So these are the related marriages. A brother and sister's daughter have married, which is, as you all know, is a first cousin marriage, and that has resulted in a horizontal affliction in a number of individuals in the sibship. So you have one, two, three, four individuals being affected, and this has not stopped there. So there is again a marriage between the third generation and the fourth generation uncle niece marriage that has again resulted in hearing loss. And as she was talking about the point mutation, this is one point mutation that has been inherited identical by descent and has resulted in the percolation across the generations. So if you look at the autosomal dominant, this is yet another mutation in the hearing loss, but in the same gene. I have selected the connexin gene so that we can emphasize on the concept that a gene can behave both as an autosomal recessive and as an autosomal dominant. That's why I said Even mere drawing of the pedigree can sometimes diagnose the pattern of inheritance. So, further moving to the molecular diagnosis, we found that the founder or the 1 4, we call this as the generation 1, and the individual is the female, the circle indicates the female. So, she is affected and she has transmitted to 50% of our children, which is a probability, but this is independent of the earlier event. So you have two individuals, two daughters who have that mutation in one copy, and you can see the small light green plus there. That means it's a normal allele. And then when this mother has again transmitted that same copy, the pathogenic allele as we call it has resulted in deafness. So this is the power of the even drawing a simple medical pedigree that will form the uh, preamble for further diagnosis. Then let's move on to the cytoplasmic inheritance, as it is called, the mitochondrial inheritance. And here also I have selected to bring more awareness to the public, a mutation that predisposes an individual to antibiotic induced or autotoxicity, if I have to use the medical term, aminoglycoside induced autotoxicity. So some individuals are predisposed to losing their hearing because of this particular predisposing mutation. As she said, A to G. Instead of A, there has been a mutation at the 1555 position. So please look at the first generation. The mother had that mitochondrial mutation, and she has transmitted to both the people, both her son and daughter, and then only the daughter would be transmitting it to the next generation, but not the affected uncle of these children. So this is the pattern which we call in genetic jargon as matrilineal inheritance. Yes, sir. I have just
Thank you. Thank you so much
supplemented with additional information.
for bringing out very clearly. So now I think we got to know about the basics of genetics, and then we also got to know about the types of genetic disorders, and then about single gene mutation. Uh, we just heard Dr. Sri Kumari explaining. The next question: what are the approaches to identifying deafness causing genes? And why genetic testing is important. The first part is to be answered by Dr. Anurujan Anand. What are the approaches to identifying deafness causing genes?
Thank you. Is my slide visible to you?
Yes, sir.
Thank you. So I'll, with the beautiful introduction to genes and inheritance patterns provided by Sri Kumari and Sudha, I'll just touch upon on methodologies to identify the genes of interest. And in this case, the genes which are pertinent uh, to the normal process of hearing. I should mention here that when we talk about a mutation in a particular gene, I'll slightly change that word. It will come handy in our subsequent discussion. I'll call that a variant. So when variant in a gene causes deafness, it doesn't mean that the normal copy of that gene, we all inherit copies, two copies, one from our mother and one from our father, are meant to cause deafness. They are actually meant to ensure the proper process of hearing or any other body function. But given their absence leads to a particular trait or a disorder or a phenotypic manifestation, colloquially we end up calling deafness causing genes. You should not be blaming genes in that manner, I must say. Now, in a genome size of 3,300 megabases, packed in 23 pairs of chromosomes, finding a particular gene responsible for the step in hearing. Uh, when I say that in the whole process of formation and functioning of cochlea from embryonic stages to the adult stages throughout our life, how is that to be pinpointed? It's a Herculean task. It is said that it's like searching for a needle in a haystack, etc., etc. And you know, there are many ways to identify a gene. Uh, there are many roads which lead to Rome, as it is said. Now, the process of positional cloning, which is the first thing I want to emphasize, is a very genetic method. I should mention here that unlike certain other illnesses, for example, cancers, where tissue uh, biological material is easily available for most of the disorders or illnesses, neurological, psychiatric, and deafness, the tissue is not available. So genetics is perhaps the best and strongest way of dissecting the genome to find out a molecule which will code for a protein most likely, and to get some biochemical grip onto the functioning of that cell. And in this case, we are talking about cochlear cell. So if we have to identify a gene in a genome, one of the method is that of finding genetic linkage. By doing what are called whole genome based studies, where you put anywhere between 1000 to 1 lakh markers. These markers are essentially DNA flag posts into the genome. They themselves don't cause any issue, any illness, so they are neutral. And we map the illness as it travels in multi-generation, multi-affected pedigrees, 2 to 3 generation pedigrees with several of their members affected. Uh, the set of markers which are present in all affected members and absent in unaffected members. There's another brand of doing the genetics. They're called genome-wide association studies, where a very, very large number of markers can be put in single individuals. It's not necessary to have 2 to 3 generation families in this case, which obviously builds up one brand of genetics, which leads to identification. By looking at sporadics and carrying out association studies, where a number of markers are very large, close to about a lakh or so, one identifies a region in the genome by eliminating 99.9% of 3300 megabases and focuses attention on a small portion, 10 to 12 megabases, and examines that further to find a variant oblique a mutation which can cause the disorder. So this process is essentially, if I have to draw an analogy, is starting by looking at the whole globe, our Earth, and coming down to a particular country, a particular state, a particular district, uh, a particular house, and in that house, a particular chair or a sofa. That's the level of resolution one has to travel in identifying a gene, a mutation. But the good part is it's very, very doable. Many people in the world are doing it. And as mentioned earlier, Dr. Ramesh, in an era which was not the most conducive to carry out these studies in the country, essentially uh, embarked on studying deafness genes in the department in Taramani. So essentially, you define a position by way of studying a large number of markers, and the markers which are co-segregating along with the phenotype of deafness gives you a way to positionally clone a gene. You need to find out a position to be able to identify a specific gene. And finding a gene is very important because you get to know what protein it's coding for. And talking in context of cochlea, for example, in context of hair cells or supporting cells, you find out what is it by way of expression of that gene that gives a specific biochemistry to that cell type. That's the reason I indicated that the basis of genes have to be seen in context of cells and structures they build up, and specialized biochemistries and biochemical milieu they create, by which we begin to call a general cell, which becomes differentiated now a cochlear cell or a hair cell. So positional cloning is one way to identify a gene, and many genes in last 30 years or so have been found, which when mutated lead to deafness. And numbers of those have been mentioned by Sri Kumari and Sudha. Now, there's another way. Um, One can look at mice models, for that matter, any animal models, where mutations can be easily generated, uh, genes and mass can be targeted, and phenotypes can be examined. I'll allude to in one of my subsequent slides what contribution such spontaneous or chemically induced mutations, approaches which can't obviously be applied among humans, have contributed to the identification of genes. And one finds homologous genes to rodent models in humans. That's a convenient way, though lengthy and laborious way, to identify genes. Um, One of the very recent methods, which is gaining force, though it was done in a very small way earlier, was to create cell type specific or organ specific, and in this case, cochlear specific library of all the transcripts, all the messages, and human genome, let's say, codes for about 30,000 such messages. We want to find out what is it that's exclusively expressed in cochlear cells. If something is expressed in a cochlear cell and exclusively expressed there, um, perhaps it has an exclusive function in that cell type too. And when mutated, could lead to deafness. So this gives another entry point into the genome, expressed genome, when one does not find phenotype of deafness in families or in rodents. And a substantial number of messages known today, which we know are expressed in the cochlea, do not have a variant or a mutation found, but are potential genes which are involved in the normal process of hearing, and when mutated, are likely to cause deafness. So as I said, one makes use of a sort of articulated combination of any of these approaches. They are not exclusive. They all feed into each other. Getting to know what is expressed in cochlear cells can help in positional cloning. Finding a gene in mice, rodents can also help positional cloning. And these are the kind of methods some of our labs in the country and elsewhere are making use to identify genes involved in this process. So this is a sort of very general perspective of various approaches which have gone on, as I said, in last quarter century to find more and more genes the way we understand and know of them today. I'll stop here and we'll
Thank
move on to
you.
other points if there are.
Thank you. I think we are just entering the complexities we have understood. And can Sudha Madam, please, can you
Yes,
say why genetic testing is important?
yes, sir, sure.
I think we all need to be moving little
Sure, sir.
faster there. Yes.
One second, sir. Is my slide visible, sir?
Yes.
Yes.
Uh, so you asked me about why genetic testing is important. So we have specific benefits out of genetic testing. The first and foremost is that it helps in precise diagnosis, accurate diagnosis. So I can explain this with an example from our own experience. Actually, a young gentleman was referred to us who had retinitis pigmentosa, visual disturbance, who incidentally had a cochlear hearing loss also. So the clinician who referred him to us was suspecting a Usher's syndrome. So initially we asked for a Usher's gene panel by a clinical exome, and he wasn't positive for a Usher's mutation. Then we went ahead with a whole genomic sequencing, which proved that he had a mutation of Stargardt's disease, and which is concerned only with visual loss, and it's not concerned with cochlear hearing loss. So, but for genetic testing, we wouldn't have had the diagnosis. So, genetic testing always helps us to have an accurate diagnosis. And secondly, it also helps us in identifying a syndrome. A syndrome is a collection of many phenotypes, but even before the additional clinical features could emerge in case of a syndrome of hearing loss, we can identify syndromes by means of a genetic testing. And it's extremely useful in making more informed treatment decisions, in the assessment of prognosis, and it also aids in making a reproductive choices. This again I can explain with an example. So we have had a hearing challenged couple, and fortunately they had a non-consanguineous marriage. And initially, when they approached many people, most of the clinicians, they were telling that both of them were congenitally hearing challenged. So they were telling that they have a high probability of having only a hearing challenged child. So when we went ahead with a genetic testing, luckily the girl was positive for a mutation in the transmembrane channel protein gene, and the boy was positive for a mutation in the myo7A gene. And both of them had inherited the disease in an autosomal recessive pattern. They had manifested the disease only in the homozygous state. So after an analysis, we concluded that they would pass on the disease only in the heterozygous state to the child. In either case, the child will have a normal protein, will not manifest the disease. We encourage them to go ahead with the pregnancy, and now to their absolute delight, they have a normal hearing child. So this was possible only out of a genetic testing. So that's it, sir.
Thank you. Thank you so much.
Thank you.
Dr. Anuranjan, can you please say how many deafness genes have been identified? This question probably will be answered by both Dr. Anuranjan as well as Dr. Sri Kumari. Dr. Anuranjan, please. And we probably will have to go little faster. Could you hear Dr. Anuranjan? Hello?
Are you able to see my slide?
The slides,
Slides,
no.
not so.
Yeah,
Yes,
yes.
yes.
Okay. Now let's get into some more details with the basics clear. You know, numbers. Our genome size is 3,300 megabases. The number of genes expressed or transcripts made in any cell type in the body uh, are about, let's say, 25,000. This number gets debated. It began when the genome was sequenced at about 40,000. Currently, it hovers around 30,000 or so. And every week, there's increase or decrease based on various parameters which we want to get into. Now, why do we want to know how many genes are expressed in relation to hearing loss? Because that tells us, we want to know that because that tells us what proportion of our genome contributes to the process of normal hearing, because eventually we want to understand how the process normally works. We want to understand it to a level so that we can manipulate it tweak it to our benefit in terms of our ability or imparting an ability of normal hearing as long as possible throughout the life, or curing it, preventing it to the extent possible. So there are 400 types of syndromic hearing loss, where hearing loss appears along with other clinical manifestations. And for a pretty good number of these, I don't know the exact number, but let's put it at about half of that, the genes are known. It has already been indicated that for human non-syndromic hearing loss, 131 plus genes are known. These numbers are give and take 1 or 2 percentage. And when we are talking about syndromic hearing loss, please bear in mind that we are talking about messages which may be expressed in cochlea, genes which are expressed in cochlea, but elsewhere too. And when mutated, can cause syndromic hearing loss type of pattern, dominant, recessive, whatever their thing. According to one estimate, there are 360 plus genes known which lead to hearing loss in humans, as well as in our closest experimental relative, mouse. Very little is known about the genes behind adult onset hearing loss. So I'm not touching numbers there. Whether the genes which are responsible for building the system, making it function early in the life, first 5 to 7 years, in one way or the other, also involved in maintaining the system and letting it function properly throughout the life, or are there exclusive set of genes which come into picture after the system gets established and now maintain it and keep on functioning, would be the ones which when mutated will cause age-related or adult onset hearing loss. It's really a major area of research at this time point in the world. We have sort of lagged behind, but this is also a very fertile area of research in the country if people want to participate in. This is essentially give us clues to find out those genes which can have the process of normal hearing go on for all the decades, 70s, 80s, because one of the goals of personalized medicine, the term gotten in by Rangasai sir, is not only to find the causes and correct them, but have a function process of hearing, in this case, function normally even in old ages. A person lives up to 80, 90 or 100 and is able to hear normally is one of the goal of personalized medicine. Now, I'm going to reflect on a recent study which came up after about 10 years of work at Sanger Center in the UK, where after having focused on human genome sequencing, this center turned its forces on looking at mouse mutants. They generated a very large number of mutations and characterized randomly without any bias, and characterized the phenotype of those, not just with respect to deafness, but many other traits and possible illnesses too. And in this study, they discovered out of total of 1,211 mutations, 3.14 percentage. Please don't worry about these percentages in decibels too much. But essentially, they found 38 genes which showed raised auditory thresholds, which represented a range of functions, transcription factors, enzymes involved in metabolism, etc., etc. To the level this study could saturate the genome and one could extrapolate the meaning of those numbers, the prediction was made that there are 600 additional genes which remain to be discovered, in addition to 362, my third line, 360 plus additional genes, 362 genes which are already known in humans and all. And this study added 38 more to that 362. So I think the sum total of my discussion is that there are as many as 1,000 genes involved in the process of hearing in humans, mutations at which can cause deafness. Um, these are very large number of genes. Each of the genes, if you connect it to the description given by Sudha and Srikumari, can have many, many mutations, can lead to both recessive uh, or deafness or both type of inheritance patterns, which are variant or mutation specific things. Now, if I try to cap this discussion, I think this close to 4 to 5% of our genome is committed to, from embryonic to adult stages, formation of the cochlea, our auditory structures, as well as its normal functioning throughout the life. And if you have to get a glimpse of genes which are associated with the process of hearing, there are all sorts of molecules up there. There's a whole gamut of structural proteins, enzymes, specialized classes of transcripts, gene regulatory elements, developmental molecules which perhaps function early in the development, obviously ion channels and transporters, cell functions, and many molecules of not well explored function, which are functioning in any of these structures, which I keep calling as specialized cell types. So you can see that there's a whole microcosmos within the cochlea, where each cell has to be given meaning, cell-cell interactions have to be established, and this done over many, many cell types with the help of many genes, as a function of time and development from embryonic to adult stages, is what creates our cochlea and auditory organs. So this is a sort of broad and approximate answer to the number of genes involved and what do they do.
Thank you.
I
Thank
think
you
I'll
so
stop
much.
here and maybe
Yes,
take
Dr.
up other things.
Anuranjan. Thank you. I think Dr. Sri Kumari, we will add whatever you want to say in the later on question probably. But I think right now I'll request Dr. In view of we are running short of time, we have many important questions also to be answered later on. So, Dr. Sudha, please. You may say how many genes have been identified causing speech and language disorder.
Yes,
I think we got to be little faster because it is almost 8:20 and we only have about.
yes, sir. Certain genetics of communication disorders. The first gene to be identified in these disorders was the FOXP2 gene. Actually, this gene codes for a protein in the FOX family, a forkhead domain protein. So that is actually responsible for modulating regions in the brain, which are involved with fine motor skills, including speech and language disorders. So any disruption of this gene causes a alteration in the speech and language development. And also this gene being a transcription factor, it affects other neural genes also. So this explains the common comorbidity of speech and language disorders with other neurodevelopmental disorders. So in addition to FOXP2 gene, we have also the CNTNAP2 gene, which codes for a neurexin protein, which affects the neural development and results in speech and language disorders. So for lack of time, I'll quickly move on to the genetics of stuttering. Stuttering again by using linkage studies as said by Professor Anuranjan sir, using a linkage and candidate gene approach in a consanguineous family, a large number of genes have been related to be associated with stuttering, and all these genes are believed to affect the lysosomal enzyme pathways. So this actually causes an alteration in the cellular metabolic level and thus results in stuttering. And autism, again, it's a heterogeneous multifactorial disorder. We have the GLP2R gene played in autism. We have so many other genes also associated with autism and attention deficit hyperactivity disorder. And we have also a group of genes causing dyslexia or a reading disability. And one peculiar thing about these genes causing dyslexia is that the variation has been not found in the gene or the exon, but the variation has been found in the adjacent regions, associated regions, non-coding regions. So that makes it a lot more interesting. And those associated regions had a regulatory expressor effect on this gene expression. So this is about it. And a lot of genes are implicated in the speech and language impairment also. So last decade has seen an explosion of our understanding of genetics of communication disorders. So for this understanding, to be translated into clinical care in a more responsible manner, and I feel there needs to be a good collaboration, a multidisciplinary collaboration between the speech and language pathologists, geneticists, and bioethicists at large.
Okay.
Thank you, sir.
Thank you so much. I think now we will move on to the question on assortative mating. Is that okay, Dr. Sri Kumari? Dr. Sri Kumari.
Ma'am, can you kindly unmute, ma'am?
Yes. Yes. So there was a lot of elaboration on the number dynamics and the projected estimates of the number of genes in the arena of speech language and the hearing loss, very well elaborated by Professor Anand and further supported by Dr. Sudha. There was one untouched field. The narrative that is going to go now is based on our experience, how we were able to characterize the dynamics of assortative mating, which is nothing but the preferential mating. So the question was, what is the meaning of assortative mating? There can be a preference for a prelingual deaf to marry another deaf. So that is here depicted in a symbolic language as deaf marrying deaf. And that is called as positive assortative mating. And it can sometimes be a choice to marry a normal hearing, a prelingual deaf preferring to marry a normal hearing. So now the question is, how does assortative mating contribute to various genetic disorders? It's not only the hearing loss, but there are other preferences in the case of dwarfism, the visual impairment, etc. But since my studies focused on assortative mating with respect to the hearing loss, I would like to dwell a little bit deep into that and what were the deliverables that we could hand over to the community. So let me start with a statement like human populations are shaped by the usual forces of natural selection, famine, disease, climate, and variation in fitness. However, there is a new force that is emerging, and this is the human culture, broadly defined as any learned behavior, including technology. So, an assortative mating among the deaf is one such cultural force, which, along with consanguinity, can have a profound influence on genetic variations, which in turn can lead to evolutionary changes in times to come. So, the marriage between individuals with the same type of recessive hearing loss, about which we have already dealt in deep and also shown in a graphical manner, are not capable of producing hearing children. So, when a marriage is between two individuals who are hearing impaired due to the same error in the same gene at the same site, or perhaps a little bit of heterogeneity is permitted within the gene, that can result only in hearing impaired children. So this is called as non-complementary mating. There is yet another type of mating which is called as complementary mating. Here, all normal children are being born. In certain scenarios, of course, despite deaf marrying deaf, both normal and deaf children can be born. This is called as a mixed segregation. It is at this point the hearing impaired families can get appropriate genetic testing and make informed decisions before making their preferential choices. This is what I want to impress upon.
In fact, this prevalence is more than 50% in the deaf community.
Yes, this is for various social reasons because the compatibility between two individuals who can sign language, or who can get along in the family, the extended family members, because other normal hearing would still know how to do the signing. So there are less number of divorces. So such are the social factors, or making these hearing impaired who go to a common deaf school. They are going to be there for 12 years, 12 long years, followed by graduation program. So they are more comfortable in communicating. There are indeed some extremities where it is called as deaf culture, notated by capital D, where they think it's just another language, the language of silence. So, coming back to this, in certain scenarios, we have done some molecular diagnostic testing with respect to a very common deafness gene. Despite the numbers given by Dr. Anand, there's one common candidate gene that is very much accountable in across the populations all over the globe. So we took up this first common gene called as connexin gene, also referred to, loosely referred as connexin gene, but it's gap junction beta 2. So let me explain this to make you understand the importance of this genetic diagnostics. If you look at the first generation, second generation, there is an individual who has one copy of the error. So she is phenotypically, or as far as the hearing status is concerned, I'm trying to minimize the genetic jargon. So she's normal. But then you can see here there is a hearing impairment, because the husband who is deceased should have carried another copy, although it's not a recessive, although it's not a consanguineous marriage. So that has resulted in the two copies in the first individual, three. I hope the pointer is also visible to you all.
Yes, yes.
So there is one individual, 3-1 as it is called, having the two copies of the variant, which is called as the pathogenic variant, and it's very well established in Indian scenario, especially in South India. But you see, there are others who have not been affected because they could be carriers. Even the concept of carrier status has been elaborated by Sudha. So that facilitates me to go forward. And if you look at this particular right hand side, you have yet another consanguineous loop that has resulted again in the connexin mutation. So, owing to the common occurrence of this particular variant, or the pathogenic mutation as it is called, you have again a marriage, although it's not blood related. They both they have preferred to marry, and they did not check upon what kind of gene is etiologically involved, and that has resulted again in all hearing impact. This is the exact thing I wanted to say. Such
Very
things
true.
can be avoided.
In fact,
Let me move on.
we have also seen a family where such a
So,
thing. In fact,
so we have counseled the such deaf matings, and also we also want to just impress upon you how a non-complementary mating can be.
yeah. Can I request you to go with the consanguineous marriage?
Yes. Just I'll move my.
How consanguinity? Yes,
Yes, yes.
yes, please.
Indeed, for the benefit of the layman, I would first like to tell you, two individuals are said to be consanguineous if they are if they at least have one ancestor in common, and it need not be too far off. It has to be only till the extent of great great grandparents, and of course an informant can inform us about how much they are related only to this level. Then this is going to happen because of the genes that are transmitted from these great great parents, great grandparents, or whoever is the founder, to which is very identical by descent. So the children born to consanguineous parents are going to be connected by both father and mother to the same recent ancestor, and can receive two copies of genes that cause a genetic disorder. So now, having discussed this, let us see what are the proportion of genes that are shared when a first degree marriage, first degree relative is addressed. It is about half the genome, half the proportion of the genome is going to be shared. And then what's the risk? It's going to be 50% risk. So whom do we call as first degree? A parent and a child, a brother and a sister, they're all addressed as first degree. Moving on to the second degree relationship, as we have seen in our previous pedigree, uncle niece. So they share about one fourth of their genome, and that results in 5 to 10% of risk as opposed to the general population. And we
So,
also have other examples under the second degree, like aunt, nephew, double first cousins, etc. And we cannot ignore the third degree. Sometimes when we go to the field, they'll say, "Nanga romba durathla related," in their language. So, but we have to be properly probing and getting those details, because that would matter to us, because 1/8 of their genome is shared. And this entails to a risk of 3 to 5%.
okay. This is irrespective of deafness or any condition.
Yes, yes.
Any condition.
As far as the recessive mutations are
Yes,
concerned,
mutations.
recessive. So diseases that are rare in general population become unusually frequent among inbred communities. This we all know. Studies confirm that consanguinity is a risk factor in recessive deafness, autism, stuttering, cleft lip palate, dyslexia, etc. So many of the reports have done incidents and all the kind of studies that would support the genetic basis in all these communication disorders. There is a considerable increase in the affected cases among parents with blood related marriages than among the control population. What we mean by control population is who randomly meet. In South India, studies are on hearing impaired from our own lab has shown parental consanguinity to be ranging from 33% to 53%. This is a very high percent as opposed to the 20% in the general population when we look at the Tamil Nadu population. So that brings me to the end of the consanguinity aspect.
No, I think, Madam, you will have to share about contributing to genetics contributing to, sorry, to stuttering.
Of course. This study was taken up in
And
our own lab, and and we are still in the process of understanding. With the help of study, I can just throw a little bit of light, but it is still in the third phase. You'd really admire that we approached about 100 schools in the span of 5 years, over a period of 5 years, and 97 schools agreed to participate. And that is in Tamil Nadu again. So we were restricting to maintain the ethnicity. And the students hail from different socio-economic backgrounds, making it more representative. Using standard protocols such as stuttering severity instrument, and in collaboration with the speech pathologists, we have a good group of that. We identified about 342 children with stuttering among the 74,544 children screened. So we can come up with, we for the first time have come up with a frequency of 0.46 that was recently reported in a journal. So this was one of the PhD scholars' work. So then our genetic studies are now focusing on the recurrent. Okay, I just want to say one more important point. These genetic studies on stuttering have shown a lot of familial aggregation and sibling recurrence risk ratios to be very high in consanguineous families. So the role of consanguinity, however, did not seem to have an acute uh, impact on the severity. So these kind of, when a disease is going to be more complex, please remember stuttering is not a single gene disorder. The Even though the genes that Sudha talked about with respect to stuttering are embedded in the lysosomal pathway. So that means these are not going to be governed by a single gene. So the role of consanguinity did not seem to be acute in with respect to the severity in stuttering. High familial incidence was, however, observed in 11% of the families that we have screened, that also point to the genetic basis, because it's still not, she has already mentioned about the mutations. So we did do a screening for the already known mutations, which she has mentioned, accounted for an overall pathogenic allele frequency of 2.3%.
Perhaps,
What
madam, you
does
should
this
talk
say?
about autism area now, please.
Yes, yes. So there is a minimal resolution as far as the stuttering is concerned. So we moved on to the third phase. And now I would like to switch the gears to tell you about the autism. So yesterday you had a sumptuous feast of the characterization of stuttering and various approaches. So I just wanted to acknowledge that and want to say that the most important change happening in the field of autism, which is considered as an autism epidemic, is to understand autism through the underlying biological processes rather than exhibit syndrome, the symptoms that are shown on the left side. So, autism is considered as a multifactorial disorder which is influenced by both genetic and environmental and immunological factors, as well as oxidative stress. That is the most contemporary paradigm that is being tested. Studies have demonstrated that inflammatory phenomenon contribute to the pathogenesis of autism. The primary being the gastrointestinal symptoms. Some studies have also suggested an association between gut inflammation and nitric oxide dependent oxidative injury. So, all this puts autism in a position which is multifactorial, which is both influenced by genic and environmental dynamics. So we just did a modest pilot study. In the case of autism, it is also very important to account for prenatal, perinatal, and postnatal stressors to understand the epigenetics or interaction between the nature and nurture. So on these lines, we did a pilot study from our lab in the phase 1, and this has supported the fact that the complications during pre, peri, and neonatal time points uh, uh, affect the neurodevelopment of the fetus and infant in the later stages that can contribute to the risk for autism spectrum disorders. So now moving on to the genetic diagnosis in autism spectrum disorders. This is also, in general, handled in phased manner. A genetic etiology can be identified in about 40% of the individuals with autism spectrum disorders. Technologies such as microarray resolve 10% of the autism spectrum disorders. Since you had a lot on characteristics of autism yesterday, I will now only focus on the genetic diagnosis of autism. Once autism is confirmed by the clinician as non-syndromic autism spectrum disorders or syndromic, we have different approaches for genetic testing. For example, the first level of genetic testing, we go for the karyotyping or the microarray for AST. When it is associated with other comorbidities, additional testing such as triple repeat expansions of CGG, which are known to be causative for Fragile X, are also investigated. Similarly, other symptoms associated with autism spectrum disorders, like Rett syndrome, we can screen the MECP2 gene and for Cowden syndrome gene. So there is always a genotype phenotype correlation, and the approach is fine tuned. These genes are the established causative genes for the respective syndromes. However, autism research has helped today to develop a gene panel that can test multiple genes at one go. In other words, it offers a middle ground between sequencing gene by gene that may be involved in the disease risk to sequencing every gene in the genome, which was also elaborated in a holistic context by Dr. Anand. To this date, autism gene panel targets as many as 53 genes. So the moment an answer is found at one of these levels, we move on to more detailed analysis of the variants identified to see how it segregates in the other members of the family, which is more simplistic, that includes both affected and unaffected, especially in the SIP. This information will also let us know whether the variant found is causative or just a risk gene. So this has to be mindfully taken. The causative genes and the risk genes. Depending upon this, the counseling can be more personalized. So I have given a layout of how genetic diagnosis proceeds from a simple testing to a more advanced approach, seeking for the answers at all levels. So one should always remember that genetic diagnosis not only answers some of the causes for autism spectrum disorders, but will lead to understanding of the neural pathways operating. So this layout in general applies for many more communication disorders that are still untread waters.
Thank you. Thank you so much, madam. I think we are now moving to a solution part. Till now, we saw various structure of the gene and ways in which inheritance is happening, how testing is done, and what are various genes that have been identified. Now, the question to Dr. Sudha is, what is genetic counseling and what do you do
Yes, sir.
knowing the gene for the disorder?
Yes, sir. So counseling is actually done in two phases. We have a pre-test counseling and a post-test counseling. And the main aim of the pre-test counseling is to tell the individual or the family the realistic expectations of genetic testing, because any genetic testing, it does not produce always a very clear answer. So we have to have a realistic expectation. And the reason why must we do the testing? So to look for the cause of the disorder, whether there are any other medical implications, to know whether there is a chance of recurrence in the family, that is the chance of risk recurrence, and what is this implication in other family members. And this is the main aim of a genetic counseling. And based on the results we have, based on the pattern of inheritance, we can tell them the probability. This we have already discussed.
Yeah,
So
okay.
for want of time, I'll skip this.
Okay, okay. The next question is, what is gene therapy and what is way forward or future trends?
Shall
Dr.
I go?
Dr. Anuranjan will
Okay.
come first.
Sir, your mic is mute.
Am I there now with everybody?
Yes, 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 yes.
Okay, so the only one point I want to mention about W24X mutation. This is in the connexin 26 GJB2 gene. While we are talking about a very large number of genes, mutations in the connexin gene seems to be the commonest cause of non-syndromic hearing loss in many populations of the world, in many places accounting for as much as 40% of all people who are affected. In our country, one of the studies we did about 1.5 decades ago in a collaborative mode, which many of the members present in this panel participated, showed that 20 to 25% of our cases can be accounted for mutations in connexin, a small gene. One of the mutations named W24X, which occurs at a 24th position, amino acid position in the gene, turns out to be a founder effect. It is not as if this particular spot in the genome in the gene gets mutated again and again. But the situation here is that this mutation has arisen long time ago in our population and has traveled through assortative, consanguineous, or any other type of matings throughout the country, in any region of the country. And without getting into details, we know that this would have arisen close to about 400 generations ago. And if you take 20 years as a generation, W24X, example of which Srikumari showed very nicely on a pedigree chart, is about 8,000 years old in our population. Now, anybody who can tell me what India was like 8,000 years ago, um, I would like to hear. I certainly don't know this because this goes uh, too far back compared to where my imagination takes me. All right. This is a large pedigree we examined in Kashmir, mutation being otoferlin in this case. You can see the number of generations and number of affected. This is a mutation which is about 5 generations old, about 100 to 125 years old in a particular region in the country. So we have many such clusters in the country which we need to learn and handle and offer some solution to their issues. While connexin 26 seems to be leading, causing 25% or so of the affected individuals, many other genes, and you can look at the names connexin 30 to TRC1, which are expressed in various cell types in this structure, which is familiar to all of you, do not seem to be accounting for more than 1 to 2%, PDS being the largest contributor in small studies for about 5% or so. So
Professor,
connexin 26 is the first gene. It
Professor,
turns out that all the known genes can be examined today, and I want to show for uninitiated people who may be curious that this is a tiny device. You can look at it carefully. It's a small chip onto which one can load millions of probes and can examine millions of variants or mutations in one go. It is like a full-fledged high-throughput lab on this tiny structure. And the backside of this, some of you may be able to see two tiny holes through which some microliter volumes of DNA can be added. And the screen that you see here is essentially a window to a small plastic slide onto which millions and millions of probes have been loaded. This particular one has 100,000 probes loaded onto which could be variant or markers or whatever. So that's the state of art with respect to thing, with respect to the technology. And one can examine not just one, but any mutation possible. Obviously, the first line of check has to be connexin 26. And if connexin 26 does not pan out, we can go on to examine rest of the deafness causing genes in devices like this. Um, these are available as auto chips. They're not population specific, but nevertheless, that's the term which is used. This is the contribution of the genes. Now,
can
where do we
we?
go from here? I think the most exciting thing at this time point are gene therapy approaches, the goals of which these are in very, very experimental proof of concept stages, the goal of which is to eventually protect, prevent or repair or regenerate the critical cell types. There are a lot of technologies which go behind it of correcting mutations, deliveries into specific cell types in the cochlea, etc., etc. And many of you who follow the literature and newspaper headlines well, the genome editing approach, which is making a lot of news by CRISPR-Cas, was the Nobel Prize winning finding of this year by scientists working in Europe and in the USA, which is paving the way towards this. And obviously, there's a long distance to go, but therapy one day is surely going to be reality. I'll give you some where gene therapy attempts in very, very experimental settings are being done using viral based, gene replacements based, gene repression based, or auditory hair cell regeneration based methods. Here, either you replace the gene, or you suppress the defective gene, or you don't worry about the defect in there and go and try to correct it at the spot, or indeed give a lot of normal protein product even if mutation exists. I should say that it's very, very important to know what the mutation is to come up with an effective strategy. And four options indicated here are just some of those options available. For otoferlin gene, there have been attempts for DFNAB 7 and 11 TMC gene, there has been attempts as one of the causes. For ushers, there's an attempt. There are for TMC1 using CRISPR-Cas methods, there's been gene editing attempted. And there are a lot of attempts in the fields of broad cell biology to regenerate auditory hair cells. And once one is able to grow these sensory patches in collaboration with ENT experts, maybe one day the day will open up to transplant these at the right places in a right manner, etc. Now, this is a long list of overview in rodent models. I think today is not the time, or it's not my
Yes.
intention now to take through each of
Yeah.
these lines, but it tells you that things have begin to happen, that it will be achieved one day in near future, and this is something our communities of experts should acquaint themselves in without worrying about how it is done, why it is done, and how successful it is, because this knowledge will embed in every profession. It permeate with medical community, it will permeate with biology community, audiology community, and there'll be contributions to be made by each one of us. And the broad opportunities that all of us have talked about are essentially shown on this particular slide, which goes from gene discoveries of various types uh, to what is current state of the art in terms of preventing by regenerating of hair cells, etc., etc., and some of the diagnostic jargon that I mentioned.
Yes.
So I think I'll stop here and we'll cover
Yes.
more as we discuss.
I do agree. In fact, Dr. Sri Kumari and Dr. Sudha may add if you have anything beyond, in addition to what Professor Anuranjan has
I
said.
think
Yeah.
Sir has
It is.
conveyed all the salient points.
Okay.
But only think
Yeah.
the mouse models, they are very promising because the ultrastructure of the mouse cochlea closely resembles that of the human cochlea. So all successful experiments in the mouse and zebrafish studies have been so promising because there are about more than 100 orthologous genes, in zebrafishes similar to humans. So all the successful mouse and zebrafish experiments holds a lot of promise. But only thing, the real challenge results in the clinical translation. So it happens if gene therapy proven, theory proven, practical. That would be a great way forward. That's what I want to mention, and especially the molecular genetics of communication disorders. When we have a careful phenotyping studies combined with it, they definitely have the potential to refine the clinical definitions of communication disorders and it to a greater extent influence their remediation. So let's not forget the fact that genetic testing is a key for future targeted molecular therapies. That's the point I wanted to insist.
Very good. I suppose this includes all the other degenerative, neurological degenerative diseases as well as
Possible.
any. Okay. Okay. Dr. Sri Kumari, do you have anything to add on this?
It's just a wrap up.
Okay. Actually, if you are adding anything to this, what is already said, you may kindly do
Yeah,
so.
I think you can move forward.
We can move forward. Then, uh, how can audiologists and speech language pathologists participate in genetic studies? So maybe Dr. Shri Kumari, you can begin. I also request Professor Anuranjan and Dr. Sudha to be prepared. I think we should go for all in scheme.
Yeah.
Can I request Anil, please? Anil, can you get all on the screen, please?
Yeah, we are all there.
Okay.
You're all on the screen, sir.
Okay,
Everyone
okay,
on the
okay.
screens.
Then,
To start with,
fine.
I think is a very professional phenotyping. If it's
Yeah,
a stuttering, it has to be phenotyped properly because that has an immense value when you get down to do more molecular studies.
yeah, I'm
So,
sure it is not only with stuttering, but every other
yeah, I'll
disorder
just,
also.
I'll cite
Phenotyping
one
is
example because
okay.
there's a lot of complexity.
Okay.
So, as genetic research in the Human Genome Project continues to provide valuable information on disease, disorders, and disabilities, it will become increasingly critical for all the specialities to come hand in hand and understand the principles of genetics. Not only the geneticists, but the other people also should understand them and make proper databases at their centers so that there's no
I
redundancy.
agree. Essentially, you were referring again to phenotyping of these conditions.
Yes.
I got
And
it.
so the core
Professor.
competencies, once they are provided, they provide a framework from which to develop genetics education.
Okay.
So competencies encourage clinicians to
Professor,
integrate knowledge
fair
and
enough,
use.
madam. I think we got that excellent point, Professor Anand.
I'm just thinking about it's a question rather than a comment or an input. Are there tools in audiology which can let us take phenotyping to a level that one can pinpoint in whatever level of resolution the defect within a cochlea by an audiological test? So I'm thinking that if the genetic knowledge can be regressed onto those audiological tests, it will not only benefit genetics, but it will also benefit a lot of functional studies.
Very true.
And one will be able to have a stronger clinical correlates, phenotypic correlates.
True.
The
I agree
toolkit of
with you.
audiology, how rich it is uh, to pinpoint that this is a defect in this particular cell.
Synoptopathic studies thus can be confirmed. And then there are other studies where one can also look at the lower brain level structures and how they transmit and how they function, and any defect in that can also be identified. But I think that this is a good point that we'll have to look at how specifically we can locate the site of lesion. So that has
Right.
been a great target in audiology.
The
Sudha
only other
madam.
point I can make for participation is that for us to, as a population in the country, to get maximum benefit of advancements elsewhere in the world, whether they are gene discoveries, we did not discover all the genes on our own. Genes will get discovered by many, many ways. But if we can put that to use in our country, and for that, it's important to have an atlas or a mutation spectrum. And that's where the community can participate, is what you asked. Um, there's one study on involving four centers in which Chennai, Hyderabad, Calcutta, Chandigarh, and our center is involved. Shikumari is involved in that, her department, which is on right now, which is going to look at about 1,000 plus families. There's a structure, specific structures. We don't want to find connexin mutation. We want to generate new knowledge also. So I would urge people to participate in that. And it's also a time point where free testing can be done as a part of this study.
Okay. Okay. I fully agree with you, madam. Sudha madam.
Yes, sir,
Uh,
definitely.
okay, you agree. In fact, I also know that there was a twin study that was happening to look at dichotic listening. Is it a genetic basis or not? Does it have genetic basis? So the twins are in larger numbers, probably in India as well, than in other developed economies. But that is yet another area where things were going on, which is fine. So what I wanted to ask you, how many genetic testing labs are there in India and where are they located? Dr.
Uh,
Anand?
okay, I can add my bit and then others can contribute. Um, at this time point, the following things are underway. Department of Biotechnology under an UMAID scheme is opening many centers called Nidan centers. These are abbreviations, let's not worry about their full forms, wherein the funding is being provided to set up substantial amount of infrastructure, either in clinical setups or in basic research setups, or a combination of these. And I'm aware of that about 8 such centers have been funded in last 2 years to a substantial extent.
Where are they
Information of
located,
this
sir?
can be found on DBT's website.
Okay, okay.
As far as Karnataka is concerned, there is a project under consideration, and the place which is taking lead in that is Indira Gandhi Institute of Child Health, NIMHANS, and Center of Human Genetics coming together.
Okay.
And they're developing protocols, also wanting to connect people digitally who can't come to these centers. And these are the centers, because they have partial funding from the government to set up, can offer rather inexpensive tests many a times free of cost if things can be managed. Some of these tests are expensive. And on other cases, they can facilitate by government funding.
Thank you, sir.
And so Center for Human Genetics is one of the places that you are aware of. It has Dr. Meenakshi Bhatt and a team which looks into it in Karnataka. And I think the overall goal is to have in every state one such center.
I think so. That's very good. And do you have anything else to add, Professor Sri Kumari, madam?
I think he has covered the government area. There are some
Okay.
corporate sectors also who provide, and the general private hospitals refer to them and get the testing done.
Okay. In corporates, actually, what is the cost? That is what we have heard Professor Anuranjan talking about free test facilities. But generally, if you go, yeah.
In our center, we have been doing a next generation sequencing, sir.
Okay.
So the cost of one test will be somewhere close to 30,000
Okay.
or 31,000 for a clinical exome.
Okay.
We do it mostly for hearing loss.
Okay. I think we will close with this question.
And I should add here that while whole genome sequencing, NGS, is very expensive, given that 25% of the deafness is due to single gene connexin mutation, testing of that can be done as little as 300 rupees or up to 500 rupees.
Also going
And one gets good level of answer for people in a testing setting. And if it turns out to be negative, then it opens up into a research kind of question, and then it becomes a little more expensive.
Yes. At least the individual comes to know that his affliction is not due to a common mutation. That itself is the important result that he can hold on and apply it when he wants to choose a spouse with the same defect. So we could resolve about 13% in this way, which could have been avoided if the simple test was done. So I recommend using this platform that, as he said, Dr. Anuranjan said, that we should use the knowledge that's already available and start screening for the simple test and account for the mutation in all the kids that are going to these special schools in the great metropolis, and then move on. For all this, we need a concerted effort, only from simple to complex effort.
Okay, I think we have tried.
Tests will be always going to field work and then convincing. So there should be a network by which we can handle this in a concerted way.
Okay, that is a good point. I think we have traveled a long way from ABCs of genes and genetics to this, and I request Meenakshi Vadera madam to take it further now.
Sir, would you like to take one or two questions, sir?
Yes, of
There are a
course.
lot of questions here. Some are, I think, must have been ordered during the process. Okay, so there was one question. There was a question, I think, in continuation in some topic about the CP children. They say if parents are fine, is genetic testing required in CP children, cerebral palsy children? It's just a general question. Probably Sri Kumari ma'am can answer that.
Actually, cerebral palsy again has a spectrum of etiology. It can be from genetic to non-genetic. So depending on the individual case, things have to be decided. There comes the personalized approach.
Like cerebellar ataxia that happens in 13 years and beyond in some families. You can get number of them from the same family.
Of course, there are implicated genes, but whether we should go directly for genetic testing or also examine other
look for other risk factors.
Yes, risk factors.
Right.
So just blindly going for the elaborate test would not be.
And there was
recommended in a cost-effective manner.
It says that it's kind of in amniocentesis, if you find a defective gene which are expressive, do you advise abortion of the fetus?
Sudha, please.
Sir, to the best of my knowledge, I feel if at all there's a finding on amniocentesis, that has to be confirmed with the chorionic villus sampling. And amniocentesis actually at present is more useful for identifying numerical chromosomal abnormalities like Down syndrome It's more useful for karyotyping like Klinefelters, Turners, and it's not recommended for single gene disorders. Single gene disorders of like hearing loss, we can do a chorionic villi sampling, provided we have to screen the fetal tissue, and we should be lucky enough to get the fetal tissue. And anyhow, that's an invasive procedure which carries a risk of spontaneous abortion. Even if it's less than 1%, we have to carry that risk in our mind. And the next question is, will you suggest for abortion? No. So ethically, it's not correct, because then the parents have to decide. And definitely, a condition like hearing loss or communication disorder is not incompatible with life. Maybe they can consider abortion in certain cases in which there are more serious conditions like cystic fibrosis and worse forms of other diseases in which the child is incompatible with life. But we cannot legally suggest abortion. It's up to them. But I think it's not recommended for hearing loss or communication disorders, because that's what I feel. And
Right.
So there's another question directed to Dr. Sri Kumari or Dr. Raghunath Rao. They say few research studies has been carried out on genetically transferred speech disorders. How far are the findings and are implemented to resolve the issues in genetic level? It's basically
Well,
looking at.
well, that field is quite primordial. There are some associations, but except for some of the genes that Dr. Sudha has mentioned, like the FOXP2, which has a great implication in verbal dyspraxia.
Dyspraxia.
Yes. Others are not single gene disorders. So we are very long way off. We have to do a proper
that's the
familial.
whole complexity of the disorder, the genetic heterogeneity.
There are a few more, but I think looking at the time, there are a lot of compliments from the attendees. I should pass that on to you. There are a lot of, word of nice words being talked. They said the thanks to you all for such extensive information, and I think we can. I think most of the questions have been answered. So I would like to thank Dr. Rangasai. But before that, I'd also like to thank Dr. Srividya from Dr. SRC Ish. She was actually instrumental in getting this panel together. It was her interest which got all of us here today. And big thanks to Dr. Rangasai for moderating it so well, and all the panelists for giving the time, their expertise. Over to you, Dr. Naika sir.
Thank you, Meenakshi. Good evening to everyone. Knowledge about genetic factors helps in better diagnosis and early identification of children at risk of communication disorder. It also helps in prevention and probability studies based on genetic counseling. Genetic studies may also provide additional possibilities for the treatment of communication disorders and gene therapy. Today's this session has thrown light on a lot of information regarding role of genetics and hereditary factors in different communication disorders. On behalf of Executive Committee of ISHA, I convey my sincere thanks to our beloved moderator, Professor Rangasai, and the panelists for helping us to clear many of the doubts that remain as a riddle even for scientists. To the best of my knowledge, Professor Rangasai is one of the first audiologists to have been involved in genetic study of hearing loss in India. Thank you for your contribution and participation, sir.
Thank you, sir.
My special thanks to the entire team of eminent panelists, for Professor Sri Kumari, Professor Anand, Dr. Sudha Maheshwari for their enlightening discussion and enhancing our understanding of the role of genetics in speech, language, and hearing disorders in an easy way. I thank you all. Signing off for today. Good night.
Thank you, sir.
Thank
Thank you,
you.
sir.
We also
Thank you
wish
so
to
much.
It's an absolute
thank.
pleasure.
Thanks to Isha and thanks to each one of you.
Yes,
Thank you,
yes.
sir. Thank you
Same
so
from
much.
my side also. Thank you. Thank you so much.
And just before we log off, tomorrow we have a very exciting panel on cochlear implants. Right, we'll be covering right from childhood to adult population, and it's all about why, when, who, and how of cochlear implants. Please join us tomorrow again. Thanks to one and all. Good night. Thanks to all the eminent panelists.
Thank you. Thank you.
Thank you.
Thank you to
Good night.
all. Thanks to all. Thank you. Bye.